We can go ahead and get started.
All right.
So I think Kathy will introduce.
Okay.
Steve, I'm going to introduce you. So today it's my pleasure to introduce Steve Goodman. Steve is Associate Dean of Clinical and Translational Research and Professor of Medicine and Epidemiology at Stanford. He directs Stanford's training programs in medical research methods, sponsored by Stanford's Clinical and Translational Sciences Award, and serves as Chief of the Division of Epidemiology. He is co-founder and co-director of the Meta Research Innovation Center at Stanford Metrics, a group dedicated to examining and improving the reproducibility and efficiency of biomedical research. He's active and plays in advisory roles in Patient-Centered Outcomes Research Institute, PCORI, the National Blue Cross Blue Shield Technology Assessment Program, and as an associate editor of the Annals of Internal Medicine. Steve's research concerns research evidence with an emphasis on Bayesian approaches. I'm very excited myself to hear his talk today, and he's going to be discussing issues of research reproducibility and focusing on the concept of cumulative evidence. So Steve, please go ahead.
Sure. Thank you very much. And I'm actually going to be talking about a very broad range of issues having to do with this thing called research reproducibility. The cumulative evidence I will definitely touch on, but I probably won't give adequate coverage to almost any of the issues here because it's such a broad landscape. So I hope that folks listening will not hesitate to either on the chat or maybe in the room ask questions if that's possible. Certainly we'll leave time at about 15 minutes at the end for you to ask to amplify many of the points that almost certainly need amplification. So let's just start off with these sets of issues which we'll be talking about today, which is why are we worried about research reproducibility, or actually what we're worried about is research non-reproducibility. What it is and is not, what causes it, and what we can do about it. That's the landscape we're going to try to navigate, and here we go. So this is a figure that shows journal articles across all of science that mention the term, as you can see in the caption on the right, either research reproducibility, reproducibility of research results, any sort of combination of those words. And you see from the 70s now to this ends of 2014, a really dramatic increase in the articles that mention this across the scientific spectrum. And in the pale green here, this is clinical medicine. But I want to make the point that this is an issue that affects all of science, and there's a huge amount of dialogue going on in many, many different fields about how to address this issue. And we'll be touching on that a little bit. But there's no question that it's getting a lot of attention these days. In some cases, it's some of the same issues that we've been paying attention to for 20 or 30 years, just given a different name. Okay.
Just click on the screen.
Yeah. There we go. So just to show you that this has been going on for a long time, this is a bunch of things identified as cancerous. This is back in 1992. Electric razors, broken arms, but only in women, fluorescent lights, allergies, breeding reindeer, being a waiter, owning a pet bird, hot dogs, being short, being tall, and if you don't fall into any of those categories, having a refrigerator. So I'm sorry I can't be in the room with you. You're supposed to actually laugh at that. But the problem is that each of these are actual papers. Real papers with real results that maybe look to somebody like they're convincing. And this sort of thing is occurring right to the present day. This is a paper that I really enjoy, written by my colleague and co-director of the metrics, John Ioannidis, and he did a systematic review of the 50 most used ingredients in the Boston Cooking School cookbook. And this is what they came up with as the spread of relative risks. These are the ingredients down here. I'll show them blown up in a second. These are the cancers they looked at: breast, GI, GU, gynecologic, head and neck, lung, other. This is the spread of relative risks. Here you see 2, 5. So, GI, big constellation of risk factors, of risk due to the cooking ingredients. And this is the blown up examples of the cooking ingredients. So we have wine, tomatoes, tea. What you see is they're scattered all over the place, from highly protective to highly dangerous. And the problem is that each one of these, or a vast majority of these, generates a headline, and it goes out to the public. Here's the sort of headlines that we might see. Well, this is for a device. Magnets lessen foot pain of diabetics, a study finds. It's probably an FDA-approved device. Down here you see a finding that runs counter to many previous studies. Well, this should be a red flag that maybe this isn't true, but apparently that's what makes it newsworthy. And I want to point out that it isn't just that it makes it newsworthy. Sometimes these sorts of things make the journals more interested in them as opposed to less. And these are quotes directly from the article. We have no idea how or why the magnets work, but it's a real breakthrough that while the study must be regarded as preliminary, so they have the appropriate caveat, but the early results were clear and the treatment ought to be put to use immediately. So again, we see a finding that looks through our eyes, hopefully to your eyes, like something that's probably a little sketchy. The theory of the magnets working on the foot all by itself is probably not very plausible. And then you have that many prior studies show that it didn't have an effect. So the question is, why does this get promoted and by the investigators like this and then picked up by the media? Now, to get a little bit more serious, many groups have put reproducibility issues on their own radar. This is Francis Collins, obviously the head of the NIH, and Deputy Director Larry Tabak. And we'll talk a little bit more about what they said, and this was from 2 years ago, where they put it at the top of the NIH priority list as one of their concerns. So why are we worried about it? Well, here's just one of many articles, and I apologize if these are old news to some of you, but to most audiences I talk to who haven't been living in this space, these articles they're not that familiar. So this is a piece written by Glenn Begley and Lee Ellis, where they were working in industry, and they were noticing that there were a lot of non-reproducible results. Sorry. And what they did was they tried to bring inside the company, they chose 53 landmark preclinical experiments, and they tried to replicate them. And they worked extremely hard at this, and they report only being able to replicate 6 of the 53. Let me show you a quote from the article. It says, when the findings could not be reproduced, an attempt was made to contact the original authors, discuss the discrepant findings, exchange reagents, and repeat experiments under the author's direction, occasionally even in the laboratory of the original investigator. So this was not just a one-off efforts casually done to repeat these experiments. This was a really serious effort to engage and get everything as right as possible. And here's just an anecdote. It says Begley met for breakfast at a cancer conference with the lead scientist of one of the problematic studies. He said, we went through the paper line by line, figure by figure. I explained that we did their experiment 50 times and never got their result. He said they'd done it 6 times, got this result once, and put it in the paper because it made the best story. Well, I probably don't have to explain why that's probably not the best way to do science. And yet, in fact, that is how science is done in many settings. Not because somebody is trying to perpetrate a fraud, but because they interpret the time that they got the significant result as the time that the experiment worked. And they will say, in this case, the other 5 times, that showed that they didn't get all the parameters correct. And so they don't not report it to deceive per se. They don't report it because they think that the 5 are deceptive and the 1 was right. And in this case, I would say they probably didn't have a sense of the play of chance. And we'll be talking a lot more about this. This is another very similar paper done by investigators at Bayer. And this is what they found. Their company, before they invest sometimes millions of dollars in developing targeted therapies, they try to validate the targets. And I'll show you the text. It says they went to the heads of their various divisions and asked them how often their attempts to validate exciting published work was successful. And as you see here, it says they collected data from 67 projects in 23 labs. 47 of the projects were from oncology. It says the analysis revealed that only 20 to 25% of the projects were the relevant published data completely in line with our in-house findings. In almost 2/3 of the projects, there were inconsistencies between the published data and in-house data that either considerably prolonged the duration of the validation process or in most cases resulted in termination. So these are the kinds of papers that are coming out, and we could spend the hour just on this issue in the preclinical space or in the clinical space. But I'm just giving you examples of the kinds of things that make people very, very worried. They speculated on why this was true, and this wasn't the result of an in-depth investigation in this case, but they thought it was due to a variety of things which have been repeated and confirmed by many others. Incorrect or inappropriate analyses, small sample sizes, poor design, poor reporting, poor control of the experimental conditions, bias towards submitting or publishing positive results, the inability of the peer review system to detect these kinds of problems. We often talk about the importance of peer review, and it is important, but there's certain foundational and fundamental problems that it can never pick up. And they also noted that many rejected papers are republished without change. So peer review is imperfect in that it only changes a paper changes a paper with respect to one journal. It doesn't work across journals. In an attempt to get a handle on this, there's been a group called the Reproducibility Initiative, and they've started doing large-scale replication projects. Some of you may know about this, in 2 fields, in psychology and in oncology, in cancer. The cancer results are just coming out now, but the psychology results, obviously a very different field, were moderately dramatic, and in some ways dramatic in ways that I think weren't really conveyed in the press. But this is a figure that shows their results, and let me just interpret it for you. On the left, what they did was they took 50 studies, I believe it was 50, might have been 100, that were published in the top psychologic journals. And they tried to run replications as close as they could to the original study. And on the left here, you see the p-values of the original study. So here's the magic p-value of 0.05. And here are the p-values of the replications. The width of this is proportional to how many of the replications had that p-value. So what you see right away is that almost all the original studies were statistically significant, and very few of the replications were. This was the median there. This is the actual effect sizes, and given that they were almost all statistically significant, not surprisingly, virtually all the original effect sizes were moderate to large, and virtually none at 0. And these are the effect sizes of the replications, a very, very different distribution. Now, We'll come back to this later, but one thing that was lost in the communication of these results, which made a big splash in the media, was on the front page of the New York Times, was this, which was that if you showed me 100 biomedical studies and 99 of them were statistically significant, I would know you had a biased sample. So right off the bat, what we see is that there's a profound publication bias in this literature where they obviously don't publish or don't accept studies that are not statistically significant. So a large part of this is just regression to the mean. They're probably picking up the largest, perhaps most implausible effects, and the negative ones are either not being submitted or being published in the second-tier journals. Now, the FDA has been looking into this in many, many ways. This is just one that just came out, and maybe many of you are familiar with this. I think this is a fantastic and really, really interesting report. And what your colleagues have done is looked at 22 case studies where phase 2 promising phase 2 results did not cohere with the phase 3 results. This is the table of contents, and I'll just point out the categories. Phase 3 trials demonstrating lack of efficacy, where in phase 2 it was shown to be promising on the efficacy realm. You can see the list of drugs. We're not going to go into this. There's one where the phase 3 trial demonstrated lack of safety. And here, this category, which is 7 products, where there was lack of efficacy and lack of safety in the phase 3 trial in something that looked promising in phase 2. Here were some of the conclusions. It really wasn't the conclusions were not that detailed, but it said these cases show that phase 2 data don't necessarily predict the product's safety and efficacy, even when the product is already approved for a related condition, and phase 2 data seem promising for the second condition. It says in several of the cases here, the experimental product was already approved for one condition and seemed promising for a related condition, but full testing failed to show that the drug was effective and or demonstrated that it was dangerous for a related condition, which is really quite interesting. At the end, they say this array of unexpected results from phase 3 studies demonstrates the complexity of the interaction between a medical product and the patient and how logical presumptions without corroborating clinical evidence can be unreliable. Now, this doesn't show that, in some sense, non-reproducibility, but it shows that if we don't have the very highest quality studies, that it's very, very possible that they may turn out to be not true. And we're, again, going to explore that in more depth. I want to talk a little bit about one lesson that has affected a lot of policy and perceptions and reflect on a few of the lessons that were learned from this. And this was the sort of debacle that occurred at Duke starting about a decade ago with respect to some incredibly exciting results that said that we might be able to use genomic gene expression signatures on tumors to guide the use of chemotherapy. And this is one of the articles, one of the many articles, some of which won awards. This appeared in Nature Medicine, and they appeared basically in every major top flight journal in the world. This is some of the figures from that paper. What you can see is this is docetaxel resistant lines. They were doing this on cell lines. Docetaxel sensitive lines. You see there's almost a complete separation in the heat map of the gene expression here. You see complete separation here, separation here, separation here. Very, very exciting results. And what's very interesting about this example, for those of you who don't know it, is that one of the reasons that we ultimately found out that none of this was true was because the investigators were very transparent. They published all of their data on the web. Here's another paper, New England Journal. Very, again, hailed as a breakthrough at the time. You'll see at the top, this article has been retracted, as have almost all of them. So just to very, very quickly summarize, and I apologize for those of you who know this. This started, these papers started being published in 2006 and 2007, and because they were sharing the data to MD Anderson bioinformaticians, Keith Baggerly, Kevin Coombs, they were being badgered by their investigators who wanted to use this. So they said, well, fine, let's go look at the data. And what they found was that I won't go into all the details, but the data had been misanalyzed. And very, very serious mistakes had been made. They were able to reproduce the incorrect findings, and after making the adjustments to show that it was all just complete noise. So they started going on the road and showing this and trying to get the journals to listen. They wrote letters to every journal. Some were rebutted by the authors, but many journals said one famous quote, I believe this was from Lancet. I apologize, it wasn't, but they said it was an argument between statisticians for which there is never a right or wrong answer. In 2009, they maintained a website where they showed all this, and they published a 120-page article in the Annals of Applied Statistics outlining all the errors. Now, I don't know how many of you are up to date on your reading of the Annals of Applied Statistics, but I would forgive you if you hadn't quite paid that close attention. It wasn't probably the best venue to rebut it, but the point was that they had the goods here, and they knew why it was wrong, and yet the systems of scientific correction weren't working. And in fact, on the basis of these results, the Duke investigators started clinical trials. So they actually started treating patients on the basis of this. Well, finally, in July 2010, everything came tumbling down because of a cancer letter revelation. What was that revelation? It was something they found on the CV of the first investigator, which is that he wrote this, Rhodes Scholar, Australia. And this was misleading because he was never a Rhodes Scholar, and it was unclear what this meant. And I won't go into all that stuff, but the point was that what led to the unraveling of this story was something completely unrelated to the science. The science had already been established to be wrong, but it was this issue that produced the loss of trust in the results, and it produced a whole cascade of events that ultimately resulted in the retractions and a major report of the IOM on the lessons from this and the standards that should be used in translational omics. Of course, if 60 Minutes got in on the case, this became quite a scandal, and from 2012 till today, most or all of the papers have been retracted. The trials are halted. There's now major lawsuits from the patients, and several of the involved journals, including Nature Medicine and Lancet Oncology, are revising their procedures about handling of letters. So what actually also interesting in this, and again part of the lessons, if we're thinking about how do we make sure that the results coming from the lab or coming from the clinic are right, there was a medical student that actually raised concerns during this, and his complaints were essentially quashed. And it's only from the inside sometimes that you know that things are going wrong. So again, it goes to the issues of policies around scientific self-correction that we have to pay serious attention to. So what were some of the lessons? Failure of processes of scientific self-correction at the institutional, journal, and investigator level. The importance of transparency. It never would have been uncovered, at least the scientific part, if they hadn't actually published their data. And honestly, they didn't think they had done anything wrong. So this was not an example of malfeasance or corruption or falsification, perhaps once they came under pressure, but not originally. And that, again, is another message. Most of issues around research reproducibility is not about fraud. It is not about falsification. It is about people using the methods that they're taught to use. Sometimes they're using them not very competently. Sometimes they're doing exactly what they're told. And we can run into these problems, and we'll talk more about that. So it underscored the importance of proper methods, particularly data management, the importance of monitoring, and many people looking at the original data and taking accountability for the work. There were issues of conflict of interest here, which I haven't talked about. And finally, the consequences in terms of the loss of public trust and consequences for patients were extreme. And we have to remember that these are not just academic concerns. When science, particularly clinical science, the clinical trials in this case, call into question, these have consequences for institutions and the funding of science as well. Now, this is a study done by a colleague of mine who's here at Metrics, where they did a systematic review of scientists themselves, admitting whether they had used questionable research practices, or they knew of colleagues who used them, or they know of cases of bias or fraud. And here you see, and this is a very, very common, a big difference between folks admitting that they had used questionable practices. That, again, this is not necessarily overt falsification, and versus what they know of in their colleagues. And you see here that the bars go from 10 to almost 80%, and we see this phenomenon reproduced over and over. Here's another set of his findings where the question is, how often do they use questionable research practices? In the clear bar is never. This is once or twice, gray, and occasionally is in the crosshatch. And as it goes from yourself, the people you work with, to graduate students, other researchers at your institution, and researchers at other institutions, they're always called into more question. And here we have rates of 40% for both once or twice or occasionally. And even at your own institution, rates much higher than when people are asked themselves. Here are some of the questionable research practices and the prevalence. And some of these are very soft, and some of these are less soft. Failing to report all dependent measures. Depending on what that is, that can actually be a major issue. Not actually reporting all your analyses. Collecting more data after seeing whether results are significant. Failing to report all conditions. Stopping data collection after getting the desired result. Rounding down p-values. Selectively reporting studies that worked. This is a big issue. And here we have a prevalence estimate around 67%. So those are some of the reasons, and only a tiny fraction of why we worry about this. So what does research reproducibility actually mean? Just one second. Well, you will see that there's actually a lot of confusion about this. This was from a committee of the NSF, and this is the Committee Not on Reproducible Research, but Robust Research. And they tried to define reproducibility. And they defined reproducibility as the ability of a researcher to duplicate the results of a prior study using the same materials as were used by the original investigator. So they said it's the minimum necessary condition for finding to be believable and informative. They defined replicability as the ability of somebody else to do the same experiment and come up with the same result. And they had other words like robustness, resistance of the findings or qualitative conclusions to changes in experimental conditions, and generalizability. Is it true? If it's true in one place, is it true other places? Now, it should be clear to you that there's tremendous overlap between these terms, and one person's robustness is another person's reproducibility or replicability. And in fact, you see the same issue and confusion in the language of this policy piece by Francis Collins that I mentioned before. And this is what they said contributed to non-reproducibility. And you can see I've bolded some of the things. Poor training of researchers in experimental design, so presumably poor design. Increased emphasis on making provocative statements rather than presenting technical details. So that's the issue regarding emphasis, spin, or interpretation. Publications that don't report basic elements of experimental design, so that's non-transparency. Some reports probably the result of coincidental findings that happen to reach statistical significance, coupled with publication bias. It's another issue entirely. And then we get overinterpretation and a claim that it's significant, but it fails to be reproducible. Well, maybe they claimed it was significant because it was significant. And this is just saying that sometimes significant studies can't be replicated. And there are mathematical reasons for that. So there are lots and lots of terms that refer to this phenomenon: reproducibility, replicability. There's also repeatability, reliability, robustness, a lot of R's, generalizability. And then we get a whole cavalcade of different solutions, and we have talk about open science, transparency, and sometimes these are the catchwords for non-reproducible science, that it's not open, that it's not transparent. But really, I would like to argue that what we're getting at is truth. And we're getting at, are our findings or are our claims true? I want to point out that a finding in general is neither true nor false. It's simply it's simply data. So if I tell you that I came up with a result of 2, that's not a claim. A claim is when a scientist takes a number and says something is true and this is what we should do. So if we use as our lodestar truth rather than reproducibility, which actually we'll talk more about, is very difficult to define, we come up with more sensible arguments. Now, one of the difficulties with respect to proof and truth is that you will find that it's very different in different domains of science. So in the domains of the FDA, there are 2 very different worlds. There's clinical and population science, epidemiology, clinical research, social science, and they have very different sort of norms and criteria than does laboratory science, which includes preclinical and life sciences. And then there's field science, computational science, and then all these sort of merged science, all the omics, genomics, proteomics, economics, I know it's pronounced differently, psychology, which is something unto itself. But it's very, very important to recognize the heterogeneity in these sciences. And they have some differences other than just culture. They have differences in how strong the signals are. That is, when in clinical science or in laboratory science, when we look for a P of 0.05, that's just 2 standard errors. That's a very, very low signal to measurement error ratio. The different, so that's a very noisy and very soft criteria. Statistical methods and standards for claims are very different across the sciences, as are complexity of designs, and also the culture of replication, transparency, and cumulative knowledge. Finally, the consequences of errors are different, and this also affects what people do. So we actually wrote an article about this, and this is what we came up with, that we would retain the word reproducibility for everything, because it is used to apply to everything, but distinguish between the reproducibility of methods, the reproducibility of results, and the reproducibility of conclusions. And I'm not going to go we'll talk a little bit about these, but I'm going to go moderately fast because I see that we're using up already too much time to have a good discussion at the end, and I'm putting a priority on that. So with regard to methods reproducibility, this is about sharing code, data. This is about making the methods transparent. So somebody else could do a similar experiment. So, and this is the FDA is probably the premier agency that gets the kind of data that allows you to have this sort of insight. People who just read papers don't have the kind of access to the primary data that you have for many of your clinical studies. So it involves sharing of code, sharing of data, sharing metadata, sharing the protocols, and it involves registration for the lab. We will also have things like cell line and reagent validation, information about the manufacturer, the source, etc. We have reporting guidelines that help us report enough so you can see through the study and understand really what was done. We have all sorts of design and conduct standards. We have quality scores. And we have assessment of meta-biases, that are biases that are hard to detect, except when we look at many, many studies. So one of the first questions you have when you talk about sharing data is, what are the data? What data are you talking about? Are we talking about the actual CT scan, the abstracted data, the coded data, the computerized data, the clean data? And I'll show you an example of where this can make a big difference. So here's a figure from a IOM report from 2013 that I was involved in. And we made this point that there's the raw data on this side and the data sets on the other. And data sharing, it makes a big difference which one you're talking about. Here's an actual questionnaire that involved figuring out when folks were smoking marijuana. And you see the results of this. This person started at age 21 years. They don't smoke anymore, and they stopped at 26. Very, very clear, right? Right, okay. So this is the coding of that, 1, 21, 0, and 26. But this is another part of the questionnaire. It says, at each age, how often did you smoke? So this person says 20 to 29. Remember over here it says they stopped at 26. 20 to 29, 1 to 3 times per month. But 30 to 39, well after they stopped, 1 to 11 times a year. 40 to 49, they're still doing a little. So it says set to 99 to be consistent, and these were zeros. You would not know this unless you saw the raw data. So is data sharing the analytic data set? I don't know. Here is a report I think you heard from Peter Doshi some weeks ago in a previous CIRSE seminar, and he has pointed out that the lack of access to the primary clinical study reports for Tamiflu has resulted in very misleading claims and impressions of efficacy. And of course, the FDA has gone through this exercise itself. This is the actual presentation to the advisory panel in the Avandia case for diabetes. And Tom Marciniak pointed out, when looking at the Accord study, lots of claim discrepancies between case report forms and the actual records within the trial. This is very contentious, but the point is, when you actually look at the primary records, you can come to very, very different conclusions. And there was even debate within the agency about exactly what the best way, about whether those discrepancies meant anything. So some thoughts on this. Methods reproducibility requires consensus about what level of transparency is necessary, and data sharing, open science principles, and use of reporting standards are all part of the solution. Let's talk just a little bit about results reproducibility and go back to the reproducibility initiative. The problem here was, what was interesting, as I pointed out, some of this may be due to regression to the mean, but what was interesting here is they, in fact, even though this was a reproducibility initiative, they had no definition, operational definition of reproducibility. And their estimate of how many of the studies reproduced were different according to which criteria they used. So one criteria is significance levels. That is one study significant and the other not? This is actually highly relevant to the FDA in the clinical, in drugs and biologic, because of the nominal criteria of 2 significant studies to confirm efficacy. You don't necessarily expect to have 2 significant studies even if something works. So you can have one study significant and the other not, and still have them be highly supportive of each other. They looked at effect sizes, whether one effect size within the confidence interval of the other, whether the combined estimate of the original and replication study was statistically significant. I would argue that that is a very good way to accumulate evidence, but it doesn't tell you whether the results themselves are reproducible. And finally, they used, because none of these worked particularly well, or were perfect, I should say, each one was imperfect in a different way, they used subjective impression. So and let me just display these. Which of these results are not reproduced? So here you have an example of this result squarely on the null, P of 1.0, And here you have a significant result, P of 0.03. But when you look at the full confidence intervals, you see that these results are not incompatible at all. In fact, they have 50% overlap. So these results are not in conflict, even though by most of the criteria they use, these would be said to be not reproduced, that one of these did not reproduce the other, when in fact they're completely compatible with chance. Same thing here. This is the only one where there's actual some degree of statistical conflict. When you just have a little bit of overlap, usually the statistical test between the two is significant. But this is not the kind of thing we normally see. We normally see this, and we normally see this. So short of outright statistical conflict, the notion of reproduce, that is consistency, is not well defined. So it was widely reported in that case that only 39% of the studies replicated, that there was no accepted measure. Relatively few of the studies were actually statistically inconsistent, as you see here. They were consistent with each other. I pointed out 97% of the original studies were significant. The real finding of the study was that there was a high degree of publication bias. And reproducibility may not, in fact, be the proper paradigm for research reproducibility. And maybe what we should just be looking at is how studies add up. And this is actually what they realized when they started doing this in cancer biology. The first 6 studies in that series have just been published. It was published in eLife in January. And they point out at the end, because it was actually very, very difficult for them to make a verdict. It says it's important not to overinterpret the results. It's clear that nuanced interpretations are necessary, not black and white conclusions. It's clear that this approach to testing reproducibility remains an experiment with advantages and disadvantages. This is a very old meta-analysis of tamoxifen in breast cancer. It was done back in the early 1980s. It's actually one of the most important meta-analyses at the time, showing that tamoxifen had an 20% reduction in the death rate from early stage breast cancer. But what you will note, they looked at 21 studies. You will see that every single one of these studies, except for this one, this is the line of no effect, this is the cumulative 20% effect, every one of these studies was non-significant, resulting in a highly significant decrease of 20%. So you could say that the null finding was reproduced 19 out of 20 times. And yet, cumulatively, we show a dramatic effect. So this slide all by itself shows that the paradigm of reproducibility in terms of statistical significance is an incredibly poor one for determining the truth of a result. And that's why we have to fall back on that and not reproducibility. So here's a list of reasons why truth is a better paradigm than reproducibility, but we're getting a little short on time. So I'll just highlight one. One is the second study more trustworthy than the first? Maybe the second one, maybe the first one was right, and the second one was problematic. So the reason that this is confusing is because in situations that are slam dunks, like maybe cold fusion, or whether we can create a human being from in vitro fertilization, where you know it occurs or it either occurred or it didn't. In other words, the signal is much greater than the noise. So it's almost deterministic. In those cases, you do want to see that you can reproduce the phenomena. And everybody can agree what reproduction means, is that you see it again. But the intuition that we get from what I'll call a deterministic experiment is not easy to apply to situations where there is a lot of noise. And that is the case in virtually all clinical studies and a lot of laboratory studies. Now, just a few words about replication and truth. That is the probability that something is true. P-values don't measure it. I'll say that again. P-values only have a very indirect relationship with truth. The American Statistical Association issued a first in its 125-year history statement last year to discourage the misuse of p-values and to say that to use 0.05 as a cutoff is bad science. I think the FDA needs to pay serious attention to that. This is an example of why it's so confusing. if this is the probability of truth using Bayes' theorem, a p of 0.05, and I can't go through all the mathematics here, it's actually not that hard. If something has only a 1% chance of being true and you get a p of 0.05, it has at most now a 6% chance of being true. If it had a 10% chance of being true before the experiment with a p of 0.05, it now has at most, I want to emphasize, a probability of being true of 43%. That is, your chance of being wrong are still about 6 in 10. If it starts off at 1 1/4, you still, after getting a p of 0.05, have a 30% chance of being wrong. It's just basic statistical mathematics. And even if something's 50-50, which they often are in clinical trials, you still have a 13% chance of being wrong. Again, that's the minimum chance of being wrong with a p of 0.05. With a p of 0.01, that moves things more. I'm going to go back and forth here, see the length of the hours or more, but you see that we're still not even close to certainty. So there is a very easy and simple relationship between the probability of replicating and the probability of truth, but it's not just a function of the original experiment. It's a function of the probability that something was true before you did the experiment, and the probability that you replicate is a linear function of the probability it was true before the experiment. Again, I won't go through the math, but we don't know how to talk about this, so this notion of replication and reproducibility is completely lost on us. Actually, I'm not going to go through all the slides on inferential reproducibility. I'll simply say that this has to do with good scientists looking at the same data and coming to different conclusions. And I had a particular example I wanted to show you, but I'm not going to do it. But every FDA advisory meeting is an example of that. This is a paper by Richard Horton, who actually polled the authors of a number of studies that were submitted to Lancet. He is the editor of Lancet, and found that in an astonishing number of cases, the majority of the authors did not completely agree with what was published, but they suppressed their own those differences. So differences in interpretation of the essence of science, it can't be reduced to an algorithm, and it's not clear whether this should even be a goal. So what can we do? Well, here's an interesting proposal in the lab, just published in 2017 by Malcolm McLeod. He said that there should be a new kind of paper for animal studies. So I want to say there's lots and lots of proposals for what we should do, and I'm going to stop very quickly because I want to get some discussion. But he said that no preliminary trial should be published without a large confirmatory preclinical trial. That is, allow the initial exploratory result to be published, but only if it's allied with this much larger study. And here they have a hierarchy of the exploratory studies, this confirmatory preclinical trial, and then finally the generalizability study, which he isn't saying should be part of the same publication. This is a list of all the contributors to non-reproducibility, and it shows that the solutions have to be multifaceted. And I'll just point out that the solutions, I could talk for an hour about each one of these. It's in the hands of scientists. It's also in the hands of the research institutions, which are incentivizing the scientists to do exactly what they're doing, which is producing a lot of good science, but a lot of science that doesn't go very far. Industry, the field, and the journals have a role to play here. The funders, in what they look for as criteria for funding. Again, often it's number of papers and number of significant findings. It's scientific production, not always scientific quality. Impact factors, other things like that. The regulators and policymakers have a role to play, and the criteria that they play is set for approving and regulating. The payers, who reimburse the technologies, and of course the public. So this is a piece from the journal I work for, Annals of Internal Medicine. Started writing about this in 2007 and asking contributors to share their data. This is the website for metrics, and I would encourage you to go there to see. This is an old screenshot to see the kind of work that we do in this area, but there are many, many entities out there that are now exploring how to make research better. So I think we'll finish this because science tells me that lectures aren't just boring, they're ineffective. So maybe I don't know what the effect of this is, but fortunately I read another study that said that that all studies are wrong. So hopefully you've learned something. So thank you very much, and I hope we get some questions.
All right, so if you are a remote participant, you can just type in your questions in the chat box there. For those of us in the room, the microphones are off mute now. You can just ask your question, or I can bring a microphone to you. Go ahead.
So I'm Rosalie Bright. I'm in the Office of the Chief Scientist. And I wonder what your thoughts are about some of the undercurrents in society about how you can't believe studies or science isn't any good anyway because they say one thing and then they say the exact opposite several years later. Do you have some particular actionable
Yes.
thoughts on that?
Well, it's tremendously difficult actionably because it involves that involves the media. And when I say media, I mean media very broadly, from the medical journals, but certainly the main media that brings evidence to the public. I think that the message that we have to get across to everybody is that individual studies often are, I don't want to say wrong, but they're just pieces of evidence. But that the scientific method is still reliable, and that with enough evidence, we can usually converge on the right answer. I think we have to discriminate between individual studies, which unfortunately often get that coverage, although the science media is beginning to get a lot more sophisticated about this. And the knowledge we're getting from the convergence of many studies and many sources of evidence. And of course, prime example of that is climate science. But there's a lot of facts in medicine that we completely rely on, but they're rarely produced by one study. It's hard to point to almost a single fact in clinical medicine for which we rely on only one study. So I think it's possible for people to have proper skepticism about individual studies, but still understand that this is part of a process that eventually does converge to the truth. And that's why, again, the emphasis on cumulative evidence is so important, and the evidence on reproducibility in terms of one study confirming or rejecting another can be a little bit destructive, because then every study diminishes the trust that people have in science instead of adding to the bricks in the wall that build up our base of knowledge. So this is why the general theme of this and our point about cumulative knowledge is so important. I think cumulative science is a very different beast than a single result.
Thank you.
Hi, this is Mark Stone in psychiatry.
Hello.
Hello. Hi, my name is Mark Stone. I work in psychiatry, and I think this touches upon a paper that I'm working on right now myself, which is about what I would call kind of spurious reproducibility, and it deals with publication of studies associating antipsychotic drugs with thromboembolism. These are all observational studies, and it seems that this began with publication of small studies, case control type studies that showed a very high risk, about a tenfold odds ratios. And this prompted people to do studies in large databases where they would come and show relative risk odds ratios along the lines of 1.3 to 1.5, sometimes larger, sometimes smaller. And consequently, because in the context of these prior studies, these studies were treated as confirmatory, where I think if standing alone, they would the difference would probably be attributed to residual confounding. And part of my discussion points out that there is a lot of reason to believe there's residual confounding. In fact, if you plot all these studies, in a funnel plot, you see beautiful correlation between effect size and standard error. So, textbook case of publication bias. And so I think just repetition is not necessarily something that is by itself that is going to help us.
So you're absolutely right that if the inherent methodology is itself flawed and biased, and particularly if you have the same bias repeating itself over and over again, the repetition is not going to get you anywhere. What I'm hearing, though, from what you're saying is that good observers are beginning to understand what are the biases, what are the design flaws in these studies, and beginning to discount them. That's what we can learn from meta-research, that the meta-research is just research on research. But if nobody recognizes the flaw and it's reproduced over and over again, you're right. We will converge on the wrong answer, and we won't recognize that what the right answer is maybe for many, many years until some good, smart, bold, young scientist comes in and points out where the errors are. But if we can point out where the errors are, if we do understand the source of residual confounding and can demonstrate that, then that can be part of your, reforming your own field. Fortunately, I can't see anybody, so I have no idea what the response is.
We have a question from somebody online. Rima
Okay.
Izam asks, I would like to discuss drug safety post-marketing. Thank you. I would like to discuss drug safety post-marketing. In drug safety, we are plagued by small power. Can you comment on the trade-off of pooling, parentheses, observational data, to find a signal versus trying to replicate the finding in two different studies, e.g. pooling across sentinel data partners versus doing a data partner analysis?
So actually, this is something that I was co-chair of an IOM report on drug safety just a few years ago that the FDA commissioned. It's a very, very complex. I don't think I can answer. First of all, I'm not sure I understand completely the question, the specifics of the question. I would just say that this is usually a case-by-case determination, because it depends on the strength of the signal and the rarity, the combination of the rarity of the event and the strength of the signal, that is how large it is over baseline, and the other contributors to the event. It's very, very different if it's incredibly rare and incredibly unique, or like a Stevens-Johnson syndrome versus just cardiovascular mortality. So I can't answer the question in full generality, because every case has a different balance of time frame, strength of signal, and rarity, and each of those will produce different trade-offs, and the likely biases from the observational studies. I would say that in many cases, for safety, the observational studies can be stronger than for efficacy, because they're not affected by some of the same confounding by indication. So I apologize for not being able to answer it very well, because I do think it goes case by case. It could be answered better if we specified, is it a common side effect, or whether the signal is large or small, that is the relative signal. It can be a huge additive effect, but still be a small signal amidst the noise. Other questions?
I see some people typing online. Maybe we'll give them a couple more minutes because we are at the top of the hour.
I do have a question, but does
Sure, yeah, they work.
Hi, Shane and Lancy. I saw methods, reproducibility in your list, and a note about quality control. And I'm wondering if you might be able to talk a little bit about quality control in academic basic research labs, and maybe from the perspective of a lab director in a research-constrained environment, how do you make the choice or the case to invest in sort of quality management systems, or should you at all?
Well, the first thing I'll say is I'm not an expert in that area.
Yeah.
It's hard to be an expert in every domain of science. I will say that one of the biggest challenges in academic labs, and we're going to start making actually a major effort here at Stanford at some point to institute reproducible research practices across the university, but there's not a consensus on what those should be. The very first step has to be acknowledgement that there's a problem. If there's no acknowledgement that anything needs to change, you're not going to be able to change anything. So doing a study at the lab that shows inconsistency between either results or concentrations, or it depends what you're talking about in terms of quality control. You could also talk about control of the design of experiments or replication of experiments as a form of quality control of the science. So I don't know if you're talking about control of, contamination of cell lines, purity of reagents, that the calibration of machines, if you're talking about that, or if you're talking about the science that's done in the labs. I would say that scientists usually respond to data, and if there are reasons to believe at the most basic level that the reagents, the cell lines, the machines, there's problematic, you should probably go in and actually test them or show data from other labs that thought they were doing a good job, that they're not as good as they think they are. With regard to the science, there's just a torrent of papers coming out pointing to the problems with the kinds of small sample size, non-blinded, poor design experiments that are often done, and selective reporting that comes out of labs. And I do think that every lab has sort of different procedures that could be used to deal with that. Depends on what kind of science they're doing. So I can't answer the question perfectly because I don't know exactly which aspect you're talking about, but it also depends a lot on the culture of that laboratory and the willingness to evaluate practices that they might have used for a long time that may in the current day not be optimal.
We have one more question from someone online. Ronnie Peterson says, as a medical doctor, do you have any comment on the communication of off-label use of medical products based on scientific findings and not FDA approval?
Boy, this is a this shows you how wide-ranging this subject is. I don't, again, very good question, very complicated subject. A lot, as we all know, a lot of off-label use is based on good science, or at least science, but they haven't submitted it to the FDA for formal approval. So I don't want to, with too broad a brush, dismiss off-label use, because so much of almost all of pediatrics is off-label use, as we know. But to the extent that off-label use is based on hearsay, it's based on poor science, that's a big problem. So I think we have to distinguish between the two. I would not dismiss all therapeutics in pediatrics just because it's not based on RCTs, although I do think that is a problem that needs resolving. But it at least is based on reasonable science from adults. It's somewhere in between guessing and not. But there's a lot of off-label use that's completely ad hoc and based on virtually nothing at all. And interesting, and just like many medical practices, it becomes very hard to dislodge and very hard sometimes to even do the science once things become part of standard practice. So I know, again, that doesn't completely address all the issues. I think the secondary issue of to what extent off-label use should be promoted, advertised, and that's more in the hands of the drug companies. That's a somewhat different subject, and I'm not going to get into that right now. And again, don't claim to be an expert, but I do think we have to distinguish between cases that are based on good science and not.
All right. I think we're a few minutes over, but I think it was a good chance for some dialogue. So I'd like to thank all participants who joined both in the room and online. And thank you, Dr. Goodman, for an excellent presentation.
Thank you. Nice to join all of you. Bye-bye.
Thank you very much.